Once we've established the formation of C5 convertase and generated C5b and C5a, C5a is going to float away into the plasma and serve as an inflammatory mediator, but C5b is going to attach to the surface of the bacterial target, or whatever the cell target might be. Once we have one unit of C5b attaching to a cell surface, then that's going to increase the affinity for C6 to the same structure, or C5b. Then C7 is going to attach, and then C8. It's much like a domino effect. Once we've deposited one, it increases the affinity for the binding of the next three components to C5b. So C5b, 6, 7, and 8 all attached to and form an anchor on the surface of the bacterial cell. And then we have multiple units of C9 that then combine with that anchor-like structure. And that's what's illustrated in this drawing. So C5b attaches to a lipid layer on the surface of the pathogen. Here we're thinking possibly like a gram-negative bacterium that has an outer membrane. C6 and 7 will then attach. C8 inserts as a transmembrane protein. And now we have the multiple units of C9 that form a channel or a pore. Once we've established this channel or pore, this is again a transmembrane structure that provides an opening for the intracellular contents of the cell to leak out. And there will be multiple units of this structure that's called the membrane attack complex that will form on the surface of the target cell. It could be a bacterial cell, it could be a red blood cell. Complement gets activated on a variety of different kinds of cells, not just pathogens. We use bacteria as our usual first example, but in this example, it's not just bacteria that can get targeted for membrane attack complexes. It's any kind of cell where complement gets activated. Here you can see an illustration of the multiple membrane attack complexes that get formed on a target membrane on the left. So all those black dots are actually the channels or openings. On the right, scientists developed liposomes, or small little spherules of membrane structures, so that they could see individual MAC or membrane attack complexes formed. And you can see on the spherule on the left a side view of one of these pores or membrane attack complexes attached to this little sphere. Again, on the right, far right, you can see sort of at the 2 o'clock position the surface of one of the single membrane attack complexes on one of these little structures. So here we have an animation illustrating the formation of the MAC, or the membrane attack complex. C5b, C6, 7 are going to form a nice stable platform for C8, which serves as the primary anchor to get this structure started. We have the pore or channel established, multiple of them, the more holes the better, and then it leads to cell lysis. And again, the lysis, or the essentially rupturing of the target, could be any kind of cell, prokaryotic or eukaryotic. And that's one of the major functions of complement components, leading to the destruction or lysis of a target cell. So complement components can serve for killing or destroying cells in cell lysis. C3b can function as an opsonate to improve or enhance phagocytosis. And the components like C5a and C3a can serve as inflammatory mediators.